The actual study, one, the embryonic stem cells or the embryo-derived stem cells, and the second group are the adult stem cells. I would like to talk about this group of cells. There are some new events which show that these cells originate from the so-called pericytes. And this is a new result which we have get after study the origin of the Leydig cells. Some words concerning the pericytes. The pericytes are cells located in the wall of microvessels, and they are covered by the basal lamina of the endothelial cells. Now, very narrow relationship between the endothelial cells and the pericytes, and the pericytes are actually a component, structural component of the blood vessels. Here you can see this is an arteriole and this is a capillary which divides in different directions. The arteriole is covered by circular smooth muscle cells, and the pericytes are distributed along the small vessels and are connected with their old growths among each other or with some of the endothelial cells. One interesting fact is that pericytes can transfer into smooth muscle cells, and smooth muscle cells can transfer into pericytes. This means that both cell types have the same origin. The pericytes, Pericyte progenitors, become distributed through the whole body with the building of the blood vessels, and they are stocked as pericytes within two vascular niches. We will see what that means in form of resting quiescent cells, quiescent adult pluripotent stem cells. Schematically, this picture shows that a capillary from the central nervous system. You see a lumen, and this lumen is covered by an endothelial cells. The endothelial cells have a membrane, and this membrane divides and surrounds an additional realm, which is called vascular niche. This vascular niche contains the pericytes, although the pericytes are enveloped by the basal lamina of the endothelial cells, and the pericytes are partially producer of this basal lamina. This here is an astrocytes. This is a fact that is saying in the central nervous system. Concerning the pericytes, it's very important to note that the blood vessel system, cardiovascular system, is the first functioning organ system during the mammalian development. That means that the pericytes, which are enveloped in the small vessels, are also a very old kind, not old, a very early type of stem cells, which we will see where they originate. The pericytes is now established, possess mesodermal, ectodermal, and endodermal lineage capacity. This capacity is very characteristic for the pluripotent epiblast stem cells, also one of the earliest cells which originated during the embryogenesis. Our attention toward the adult stem cells was inspired by the studies on the origin of the testicular Leydig cells. Words concerning the Leydig cells. The Leydig cells are situated in the testicular parenchyma, and you can see here, this is the tunica albuginea, a very thick covering from connecting tissues. If we enlarge a part of the parenchyma, we can see that the parenchyma consists of different sections of seminiferous tubules and intercellular substance. And if you note, there are a lot of blood vessels in this intercellular realm. Additional, there are small or large groups from large cells. These are the so-called Leydig cells or interstitial cells, which are the producer of the steroid hormones, primarily testosterone. These cells was found by Franz Leydig, a German zoologist and anatomist, which in 1850 for the first time described this type of cells in different vertebrates. More than 100 years were left until we have become information about the functional significance of the Leydig cells. By means of enzyme histochemistry and biochemistry, it was established that the Leydig cells are endocrine cells, and they secrete steroid hormones, most of all the testosterone. During the last 2 decades of the 20th century, we were able to establish that the Leydig cells possess also neural characteristics. And we defined these cells as neuroendocrine cells. And this was also the region why we put the question, are the Leydig cells of neuron origin? And this has put us to search on the origin of the Leydig cells. We have performed a very large study with many methods, and we found a very good experimental method by which we, by only 1 injections of the toxic reagent ethan dimethan sulfonate, we were able to destroy all Leydig cells which are present in a testis. After that, in a period of 3 to 4 weeks, a new Leydig cell generation arises, and this is a regeneration which proceeds from stem cells. And that is what we have shown. We have found that the stem cells of the regenerating Leydig cells are the pericytes and the smooth muscle cells. I will talk about the pericytes because these are cells from the same origin, and not every time will I say smooth muscle cell and pericytes. Also, if I told about pericytes, I mean also the smooth muscle cells of the testis. We found during this experiment that the pericytes behave exactly as stem cells. They are located in a capillary network. After activation, they proliferate and migrate and build the so-called transient amplifying cells. These are middle cell form, which then became differentiated terminally to Leydig cells. This is the scheme which is characteristic for all stem cells. They follow this scheme to build differentiated cells. I would like to show you some experimental preparation. You see here a capillary and a precapillary here also. After intoxication with the toxic agent, a very strong activation of the neurofilament protein nestin become evident in the pericytes. And the same cells, as you can see, are the cells that divide very, very actively, and they build a large number of new cells, which cells migrate in the neighboring connective tissue between the tubuli, and you can see that from one capillary, here are the pericytes, cells divide and migrate and build different large globules. This is here also nestin. And these cells are transient amplifying cells, which divide some more to arrive the number which is necessary for a normal organ in the old kind of person. You can see that's cells in the red. Some from the transient amplifying cells divide further, and they reach then the end state of the number of the Leydig cell. We was able then in 2004 to publish these results, and we termed that we have found who are the Leydig cells, Leydig cell progenitors. Subsequently, in a few years after that, it was very, very interesting to establish in the literature that the same processes were found in a large number of organs, and exactly in all organs, all organs in an organism of the vertebrates. Here you can see schematically, graphically, that a part of our organs of an individuals are supported by the pericytes and the smooth muscle cells of the arterioles in the regeneration of this structure. You can see bone, muscle, skin, brain, heart, lung, liver, and all that all other organs I cannot all here show. Despite this strong evidence that we have found in the literature, up to date, there are some controversies concerning mainly on problems related to the identification of tissue structures that led to erroneous results and interpretations. I would like to show you some of these. The first problems concerns the origin of the adult stem cells and the pericytes. At this time, more more origins of these structures are discussed. One, the mesoderm or the mesenchymal stem cells, the bone marrow, and the neural crest. As I told, we have also found that the pericytes are a very serious part of this process. To understand what I would like to explain after that, I would like to remember you that one new organism arises through the combination of a haploid oocyte and a haploid sperm cells. It arises a zygote, is diploid, and this zygote started to cleavage. And the next stages are morula and a blastocyst. Ladies and gentlemen, it is very important to remember that at the moment in which our daughter cells derived from the mother cells, these cells fall under the influence of the surrounding cells and also from surrounding different biological environmental factors. That means that these cells started a process of differentiation, and this process is accompanied with inhibition of some and activation of other genes, which leads to changes of the phenotype, of the biology and the function of these cells. That means that one cell can go in another direction, and there started a new cell type. This we can follow in the next scheme. You can see here, this is a zygote with two blastomeres. The first difference arises in the cleavage of the one blastomere. This blastomere proliferate faster and build a large number of cells, which at the end build the so-termed epiblast cells. This is an asynchronous division of cells. This asynchronous division of cells leads also to asynchronous activation of transcription factors, and you can see that there arise different cell types depending on the situation with the genetic material. Another problem which is connected with this division is that we have in the morula and in the blastocyst symmetric radial division. This division leads to the building of polarized cells. And we have another kind of division, namely asymmetric tangential division. In this, one cells remain here connected with the basal lamina or with the zona pellucida, and the other cells migrate in the inner part of the blastocyst, and these cells are the cells which build then the so-called epiblast. At the time of the implantation, the blastocyst contains two types of cells. One other sheet of cells, this is the prof ectoderm, it is responsible for the building of the placenta. Next sheet, this is the primitive endoderm, which is called also hypoblast, and it built the yolk sac, and the most cells built the intracellular mass built the epiblast. New studies show that the first products of the dividing of the zygote are totipotent. That means that during the division and the differentiation of the cells, they reduce their possibility to be more potent. At the beginning, these cells are totipotent. Totipotent means that this cell can build a whole new organism with all what it needs. After the further division, the late morula and early blastocyst was called plenipotent. What means plenipotent? Plenipotent means that these cells can build exactly the same cells which are building by the totipotent cells. But these cells have not more the possibility to construct from these cells a whole organism. Also we have all cells, but they cannot build an individual. And in the late blastocyst, we have in the epiblast here cells which are pluripotent. They are with smaller possibility what the potence, not the whole cells, nor they are building cells which are partially built. I would like to show some graphical presentation which concerns the epiblast. The epiblast originates from the inner cell mass, and the first cell types which it built are the trophectoderm and the primitive endoderm, or the hypoblast. The next cell type which is built from the epiblast are the primordial germ cells. They are very early built and distributed to the corresponding organs. Our studies and studies now from other persons show that also the Pericyte progenitors arise from the epiblast. Also, they are a very early form of stem cells. What happens with the neural crest cells? The neural crest cells originate later, and not from the epiblast direct, but from the ectoderm. And this is the cell layer which is responsible for originating from a lot of different cell types. That means that the neural crest, I will show that in the next pictures, some word about the neural crest. You know, the neural crest is built at the border between the non-neural epithelium and the neural plate from the embryonic ectoderm. The cells move in the deep here and build the neural fold, and this fold here is the place where the neural crest originates. And we know that the neural crest cells move then through definite ways and are distributed throughout the whole organism. One of these ways is the dorsolateral pathway, and the other is the ventromedial pathway. Thus, the neural crest cell arrive all organs which are innervated by the vegetative nervous system. Also, for the skin, melanocytes are built. Our knowledge is until this moment that the neural crest cells are pluripotent and are able to produce mesodermal, ectodermal, and endodermal progeny. And as we can see now, that is not totally right, because very new results show that in the region of the neural fold, two cells built, namely two genetically different populations. One are the authentic neural crest cells, and the second are ectodermal cell or the metablast domain cells. Schematically, you can see what that means. We have here a cross-section through the neural fold in this region, and you can see the neural plate, and at the end of the neural plate here, the authentic neural crest cells arise. In the neighbor part of the neural fold arises the metablast or mesoectodermal domain. Exactly, these cells perform epithelial to mesodermal or to mesenchymal transition. And these cells are distributed in the interstitial space here in the region of the neural fold. And these cells are then responsible for regulation of the migration and the behavior of the true neural crest cells. These results show that the authentic ectodermal neural crest cells, in contrast to the pericytes, are not able to produce ectomesenchymal or the mesenchymal mesenchymal structures. This is in fact the property of the metablast cells. Also, this is a very, very important result which shows that the imagination at the moment is not this which was shown in the new time. So, additional arguments for the leading significance of pericytes as adult stem cells, I would like to show some additional arguments for this conclusion. Firstly, the location. The pericytes are located in true microvascular niches and have a periendothelial location. In contrast, the mesenchymal stem cells and the neural crest stem cells are located perivascular or interstitial. That means there are something away from the blood vessels. A lot of people is reading for all these cells that they are they are situated perivascularly. That's not true. The pericytes are periendothelial, and these are perivascular. The second problem, or the second argument, is the maturity or differentiation of the stem cell. The pericytes, we have shown, are highly immature stem cells that maintain their pluripotent or the mesectodermal and neural phenotype through the whole life. In contrast, the neural crest cells and the mesenchymal stem cells are embryonally later arising stem cells with advancing age restrictions and their lineage potentials. Graphically, this can be presented. So we have the pericytes. You see the pericytes have the whole life, are pluripotent, and are able to produce all structures which are found in an organism. The mesenchymal stem cells and the neural crest stem cells are at the beginning pluripotent, and with the differentiation, they lose part of this potential and become at the end to the unipotent possibility. Our next problem is that a lot of people is not able to watch or to registrate the very fast transdifferentiation between the pericytes and the daughter cells. Yes, you can see here that this transdifferentiation is a very short time process, 1 to 5 hours, and at the end, the resulting cells lose any similarity to the mother cells. Yes, and this is very difficult to combine or to show that the pericytes are the mother cells of the end cells because they show no similarity. We were able to show this in our experiment in the testis. You can see here a group from transient amplifying cells, and we have performed a double immunocytochemical staining. At the first, we show nestin for the progenitor cells, for the stem cells, and the cytochrome P 450 SCC. This is an enzyme which is involved in the synthesis of steroids. And you can see that we have found cells that primarily produce nestin. Also, these are stem cells. And we have cells that equally produce nestin on cytochrome C. And we have a third kind, cells which primarily or predominantly produce cytochrome C. This means that from the stem cells, which are the pericytes, Leydig, young Leydig cells are built, and they have no similarity to one to other. We have shown also at protein level, using a Western blot experiment, that this is really true. You can see here, after ethandimethansulfonate treatment of the testis, of the rats, that between the 3rd and the 20th day, no cytochrome P can be seen. This means there are no cells which are like Leydig cells. At this time, exactly at this time, a very high expression of the nestin of the progenitor cell, of the stem cell factor nestin, can be seen. And here you can see here, at this time, begin the building of the transient amplified cell or young Leydig cells. The cytochrome becomes stronger. In contrast, the nestin becomes smaller and very, very low. Yeah, very low. We have also another problem in the literature that a lot of people is not able to differentiate it between cells are situated in a close apposition. And this is interesting in so that I give here also an example from the testis. You can see here a tubulus, another tubulus, and you can see here bromodeoxyuridine positive cells after ADS treatment. And most of the people has explained this result as results which show that the lamina propria of the tubulus are the is the structure which are built the stem cells for the Leydig cells. This was the reason is why these people has used a normal light microscope. We have used here a differential interface contrast Nomarsky microscopy, which give profiles of the non-stained structures. You can see that exactly here at this place are blood vessels is seen. These are erythrocytes here in the blood vessels. And that means that these cells are not cells from the lamina propria. These cells are cells of the blood vessels. Also, there are pericytes. This is a bromodeoxyuridine dividing from cells. Yes, dividing cells take up this bromodeoxyuridine, and we can see this by immunocytochemistry. Yeah. There are exactly very large difficulties in the recognition between the endothelial cells and the pericytes. And this is visible from this example. You can see here also in the rat 14 days after ADS, we have stained this preparat immunocytochemically for the neuroglial protein 2, a typical marker for pericytes. And you can see that we have stained here the pericytes in the capillary wall. This is also, but the endothelial cells, they are negative. Also negative are the perivascular cells here, the adventitia of the capillaries. This means that a lot of people is not able to see by normal microscopy the neighbor cells, and they defined these cells as endothelial cells, not as pericytes. If we look at the blood supply of the testis, of the rat testis, this is a scanning microscopy picture from corrosion cast. The tubules are here destroyed, and we can see a very large network from vessels, from capillaries, and they lie and build a very, very tight network around the tubuli. Schematically, you can see that these vessels are the peritubular vessels which run longitudinally, and these are connected by the circular leaving intertubular blood vessels. And you see this is a network which surrounds every tubulus, and we can now see the stem cells for the Leydig cells are really product of this network, of the vessel network. Here in the Department of Anatomy in Varna, very important questions which is under study is the adipose tissue and the angiogenesis. I would like only to show that the adipocytes originate also from pericytes in the adipose tissue. We have here the pluripotent Pericyte progenitors. They reach the blood vessels, and from the blood vessels, if they are activated, they build adipoblast, and these adipoblasts produce white adipose tissue and black adipose tissue. And these tissues can change one to the other and back, and this is also a marker that these cells originate from the same progenitor cells. So, ladies and gentlemen, some general conclusions. The Pericyte progenitors, we mean, and also other still, that there are very, very aerial cells which are originated from the epiblast during the embryogenesis. The pericytes are distributed through the blood vessels throughout the whole body, and the pericytes are pluripotent stem cells similar to the epiblast cells. They possess all three kinds of progeny, ectoderm, mesoderm, and ectodermal structures. The pericells remain during the whole life within the true microvascular niches and are reserve cells for repair or regeneration of the tissues and the organs. This is a conclusion that shows the pericytes are a very impressive achievement of the biological nature. The aim of these cells is to supply every tissue and every organ with a local population of adult stem cells, which are needed for the physiological and pathological repair and regeneration of these tissues. And mesenchymal stem cells, which are at this time also involved in this process, are surely the daughter cells of the pericytes, are descendants from the pericytes, and their heterogeneity is connected with their differentiation. If such cell differentiated, he lost a lot of his pluripotency and became to the end stem cell, which is typical for this organ from which the pericytes are isolated. In other words, we think that the pericytes are the adult stem cells, and this, I hope, will be confirmed in the next future, also from other scientists.